Good morning, everybody. Today I'm just going in to check on my cells from Friday, and I'm also going to work out my molecular cloning strategy. So I've cloned in the first piece that I needed. Now I need to make more cuts within that piece and put another piece in, but I will explain that better this afternoon. Good afternoon, everybody. So I was in for a bit longer than I expected because we actually got our molecular cloning strategy together and just decided to get started with it. So I did split down my cells as I expected to do, and they're looking healthy, but then I also got to the ligation stage with my molecular cloning. So that ligation is happening right now as I speak and will continue to happen overnight. And then tomorrow I'll do a transformation. Nothing you haven't heard about before, so I won't go over it again. See you tomorrow. Good morning, everybody. So this morning I am heading in to do my transformations from my ligation last night. So again, I have chemically competent cells and I'm going to put my ligation into them. I am also going to be checking on my cells this morning, which I transfected on Friday. So I refreshed the media on them yesterday, and I'm running out of media. So I need to make more media before it is urgent. And I also need to make new media before I run out and need it, especially as tissue culture can get so booked up so quickly. I don't really have the time to stuff around and make it once I realize I'm out. And yeah, I think that's the plan for today. I will see you guys this afternoon.
So while I was at work today, I spent a bit too much time filming because I wanted to make some YouTube shorts, but unfortunately I filled up my storage. So instead, I'll talk you through my day with that footage. First things first, popping my media in the water bath to start warming up. In the ice bucket, I've got my competent cells, so they're cells that are ready to take up some DNA. They're E. coli cells. And then the 1, 2, 3 I've got on the bench are my ligations from the previous night. So in number 1, which I've just put in the cells there, is my backbone to my insert at a 1-to-1 ratio. Ligation number 2 was my backbone to my insert at a 1-to-3 ratio to give it every chance of that insert getting in. And number 3 is my empty ligation. So that is a plasmid that should not be able to close up because I didn't give it the insert. So then we have a half-hour wait period. In non-COVID times, I would grab a coffee, but seeing as I couldn't have my coffee, I decided to keep working. So I'm just checking out my cells. When I'm checking out my cells, what I'm looking for is no contamination, so no fungal growth or bacterial growth, but also that they're growing nice and evenly across the plate. The 6-well plate is the transfections that I did last Wednesday, and the larger plate is just a reserve of cells that I'm keeping for my next lot of transfections. Okay, 30 minutes is up, so I am heading down to the bug room to heat shock my cells for 45 seconds at 42 degrees. That's going to really stress them out and cause them to open up their membrane to take in DNA that may help them survive this critical threat. Okay, 45 seconds in the water bath, and then we put them on ice for 20 minutes to let them chill out a bit more. And I'm just going to add in that nice warm media that I warmed first thing in the morning to give them some growth media. Make sure the lids are on really tight and sticky tape them up, and then we're going to pop them in the incubator, shaking for an hour to let them grow up a little bit. All right, an hour later, we are grabbing them. Probably should have left them in the incubator while I labeled all these plates, or ideally thought about this ahead and labeled these plates during that hour. But that's okay, I'm in a new environment, getting used to the protocols here. So on these plates is my initials, the date, and which ligation is in which plate. Lighting the spirit burner is part of sterile technique that is keeping my area clean and making sure only the bacteria that I have put into these plates grows on these plates. So I'm using 2 plates per tube because I found that they were a bit too wet last time I did this. So by splitting the same amount of liquid across 2 plates, each plate will dry a bit more quickly. And then using my glass hockey stick, which is what we call basically a bacteria spreader, using that to spread my bacteria across the plates. The idea here is to spread the liquid out so that it dries, but also I want my colonies to grow quite distinct from each other across the plate. In between different bacteria, I'm putting that hockey stick back into ethanol to sterilize it, and then also burning it off on the spirit burner before I use it on my plate. I really want to spread it out into the edges so that those colonies that are going to grow as far apart as possible. And that is when my phone ran out of storage. So this is pretty much what I did today. You can't see me put it into the incubator afterwards, but it just goes into the incubator at 37 degrees overnight to grow those colonies. And then tomorrow morning I go in and see if any colonies grew. See you guys tomorrow.
Good morning, everybody. It is nearly 7 a.m. So today I am going to do the third transfection. So I don't have the results from the second transfection yet. So the second transfection is still growing and I'll be doing the FACS on that tomorrow. But this overlapping of experiments is quite common in science. Otherwise, doing something 3 times that takes more than a week would take more than a month. I've also got my transformations from tomorrow to go check on. So I'm hoping some colonies have grown on that plate from yesterday. If you want to see how I did the transformation, check out my shorts. Other than that, I'll see you this afternoon. Good afternoon, everybody. Very productive day. I did a round of transfections. I got my colonies going for a mini prep tonight, and I'm all ready for FACS tomorrow. Tomorrow is going to be an even bigger day, just in terms of number of things I need to think about. I need to change over the media from my transfections today. I need to be ready for FACS at like 1 o'clock, I think. And I'm also doing the mini prep from the overnight culture. So mini prep, getting plasmid out of my bacterial cells, which are growing right now. Yesterday, my phone storage completely filled up, and so I couldn't film in the afternoon. Whoops. I'm getting a bit better about phone storage now that I'm filming this much, but it's not quite a habit yet. See you all tomorrow. Good morning, everybody. Goals today are a mini prep of my cultures from yesterday and FACS at like 11. Very excited to do that again. See you this afternoon. Good afternoon, everybody. It is Thursday afternoon. It's a bit of a chilly breeze, but there's some sun. I nearly didn't film this afternoon because today was pretty disappointing. I got my FACS data from my second round of gene editing, and it just looks like I haven't edited any of my cells. So whether that's because I'm not that good at FACS analysis yet, and I need to do some more work on that, that's possibly the answer. But from what I do understand, it looks more like I've just not edited any of my cells. So that's not a gRNA problem, it's just a mistake. So yeah, something funky going on with my FACS data or with my gene editing, not really sure which. And on top of that, I did a mini prep today. So mini prep meaning I'm taking plasmid out of my cells, and the concentration of plasmid at the end was just really low, about 10 to 20 nanograms per microliter each, when it should be in the hundreds. So again, I don't know why. As far as I recall, I did every step. Nothing seemed off at the time. But yeah, not really sure about that one. Although I did knock over 1 tube at one point, which I don't think I've ever done before. So that doesn't feel good all on its own. But nothing happened that would lead to these low concentrations. So not a great day. I almost didn't film this because I just felt so down on it all. And I just don't know why any of those things have happened. Hopefully I will work some of them out in the coming days and weeks. But for the moment, things aren't working and I don't know why. Anyway, I'll see you guys tomorrow. Good morning, everybody. It is a new day and I am ready for it to be better than yesterday. So this morning, all I've got to do is split down my cells for the weekend, which is a pretty typical Friday activity. I don't think I'm going to get much of a chance to go over my FACS results with my supervisor before the lab meeting though. So I might not get any resolution to that today. That one might have to wait for next week. We'll see how it goes. Good morning, everybody. Lab meeting was actually quite long today, so I got to hear a lot about what everyone's up to. My section was quite short though. I was mostly just explaining that my FACS indicates that my transformations didn't work. People did have some different ideas about where my FACS analysis could have gone wrong or where my transformations could have gone wrong, so I will have a look at those on Monday. But in the meantime, I'm feeling a bit better about everything. I'm just going to enjoy my weekend. If you do enjoy this content, remember to subscribe, leave me a comment and tell me what you like about it or what I could do better. Other than that, I will see you next week.